So good morning, everyone. I'm very thankful to Amber and all the people. I'm so glad to see old friends and new friends. So I'm definitely an old friend. I've been here for fifth time. I start to work on SLCs in 1A before Maxwell probably is conceived. Yeah, so today I would like to give you very brief outlines about what we have done before, especially for the new friend. I will give a brief summary of our previous work, and then I will have a brief summary of the major molecule phenotypes in two mouse models. Last one, I will talk about brief neural behavior phenotypes in these two mouse models. So as I said, we have studied in cell model first. So in previous talk, I talked about the work in cell models. We have looked at about 60 mutations in this gene, and you can see we have published most of our work. We have a look at this mutation from various perspectives. To summarize our previous work, so we want to say a majority of the mutation, if not all, they cause loss of function, can be partial or complete loss of function. Mutation can cause destabilized protein and reduce cell surface expression. This mutation can cause increased ER retention of the mutant protein in both neurons and astrocytes in both human and mouse cells. And we have done EEG recording. We identified absence seizures in both mouse models. It's consistent with the talk with Vinny and John. So we also identified a drug for phenylbutyrate can restore GABA uptake. So at least partially rescue the functionality of the GAT1. And this is already in clinical trial. We are very happy for that. So in both mouse models, we have identified the drug can mitigate the seizures and improve neural behavior phenotypes. So just give you a very quick idea about how this mutation affects the functionality of these variants. So we have compared with the wild type with GAT1 inhibitor, CL966 and NEC711, and also we compared with the MOC amplifactor, pcDNA, and with multiple variants. So from here you can see across all these mutations, they have a very clear reduction, but the extent of reduction is different across these mutations. So I would like to let you know the trafficking pathway of this GABA transporter. So this is from a paper we published years ago in JAMA for GABA A receptor, and we identified the pathway of GABA transporter is very similar to GABA A receptor subunit. So I use this cartoon. So first they have to synthesize inside the ER, the ribosome, and then they have to move on to Golgi and then endosome to membrane and then synapse. So only when they reach the cell membrane and synapse, they have a function. So this, they have to function here. So because GABA A receptor only expressed in neuron, but the GABA transporter is expressed both in neurons and astrocytes. So we look at both astrocytes and the neurons. So this in mouse, the live cells. So here, for the wild type, you see very nice, even distribution of the GAT1, which is tagged with OFP, in both astrocytes and the neurons. However, for Maxwell's mutation, you can see you don't see much signal around the edge, but you see very enriched signal around the nuclei. So this suggests the mutant protein is stuck inside the cells. So this common pattern is observed in both astrocytes and neurons. So in neuron, you won't see as clear, but I think that the subject to same mutation. And now we also did a study in mouse astrocytes to see how this mutant protein oligomerize with the wild type allele. So in wild type, this normal astrocytes culture from mouse use the red type, the green is overexpressed GAT1, and then the red one is endogenous GAT1, and the blue one is the nuclear marker. So we want to see if the mutant version and the wild version can interact. So from here, we can see the wild type, you see both green and red have a nice distribution at the cell, at the membrane. But for these two different mutants, but mutant will form very large clump inside the astrocytes, suggest probably the mutant protein is stuck inside the cells. We also look at Maxwell's cells. So we use his cells and also CRISPR-corrected isogenic control. So we studied from iPSC to neuroprogenitor cells to inhibitor neuron to enlarged. I saw this morning like a presenter anyway from iCell show very similar morphology of what we have seen here, the inhibitor neuron from Maxwell cells. So this is enlarged. So basically we reprogram from iPSC onto neurons or onto astrocytes, depending on which experiment we want. So this very nice inhibitor neuron and also astrocytes. These are all F cells just before we do experiment. This is the enlarged version, so you see. And then this is the staining, the fixed cells. So this is Maxwell's CRISPR-corrected cells. You see very beautiful, like puncta, kind of yellowish, which is co-localization of synapsin, the inhibitor synaptic marker, and GAT1. However, if you compare Maxwell cells here labeled as a patient with corrected, you see reduced puncta. If you look at the somatic region, which is marked like here, the GAT1, actually the fluorescent signaling is very similar. So we think the protein is mainly lost in the non-somatic region. And then we also look at the astrocytes. As I said, we know the gene is expressed in both astrocytes and neurons. So we found out the expression level of GAT1 in astrocytes, the human iPSC derived is much lower in neuron, but it's detectable. So you see, compared with the isogenic control, Maxwell's astrocytes have reduced GAT1. And then we use his isogenic control cells, we overexpress either wild type or his mutant version. And then compare with the wild type, you see, you have a pretty nice, like even distribution at the membrane, but Maxwell's mutant protein, you can see, is very enriched around the nucleus. This is more like a classic ER intention. So this just shows some example. And then we wonder, where is the mutant protein? Can it reach cell surface? As I showed before, just like a GABA-A receptor, GABA transporter has to function at the cell membrane. So we use a high throughput flow cytometry. So here you can see we sampled 10,000 cells, viable cells. The blue and the cyan here is either control or W193X, which is a mutation we have looked at extensively in lab, which is loss of function, has no protein expression. So we also use this as control. So you see overlapping signaling of untransfected control versus the mutant without protein. The wild type, the red one, you see nice presentation. And then you quantify, you see, compared with the wild type, if you take as a 1, and untransfected, or we use a pcDNA, or sometimes just like a mutation, like a W193, you see reduction cross mutations. This is for surface. We also evaluate the total protein. It's very similar, the pattern is very similar to what we see at the cell surface. However, some mutation, you see reduction at the cell surface level, but at the total level, they actually is very comparable to the wild type. And then let's move on to the mouse model. So we have a look at A288V and S295L. So we choose A288V because this is a common mutation identified in multiple cohorts. This has been identified in China, in Israel, in Europe, and also in America. It's Charlie, right? Charlie's mutation, and this Maxwell's. So we choose this one, these two, because they represent a different thing. So A288V represent a partial loss of function in our hand at least. And Maxwell's represent a complete loss of function. So here we have a very detailed comparison of the function at the molecule level and the functionality of these two different mutations. So this is wild type again. This is the wild type, the cells express the wild type, but treated with GAT1 inhibitor, CL966 and NEC711. This mixed wild type versus mutant of A to the V and S25, you see reduction, but not as much. But if you transfer the mutant alone, A to the V, about 25 to 30% function remaining. But Maxwell's, you have none. So this is why I said A to the V for Charlie's mutation is a partial loss function. But for Maxwell, it's complete loss of function. And now look at the protein level, because we said we wonder how much the mutant protein can travel to cell surface compared with the wild type. U stands for untreated, unprocessed. H stands for NH, you specifically remove the glycan attached inside the ER. So if you remove this glycan, if the protein can be processed, that means there's retained inside the ER. So compare with A to A V, the Charlie's mutation versus Maxwell's mutation, we see like maybe 30% high band here compared with the wild type, but the Maxwell's mutation, you see none. So it's consistent what we see here. You have no function, but here you have some function. So everything, I think, holds true. And now, how about look at the mouse model? And we use litmates. So we have Charlie's and Maxwell's. This is all gender matched from same litter, compared with the wild type and heterozygous, homozygous. We look at all different brain regions, cortex, cerebellum, hippocampus, thalamus. We see cross different brain regions, we see global reduction of GAT1. However, we see some in homozygous, in Charlie's mutation. This, again, suggests some protein is functional. And if you look at, still remember what we talked about in cells, you have about 25 to 30% protein is functional. But Maxwell, you have none. So this why in homozygous mouse model, you have almost nothing. So this why I think everything we've done so far, that's consistent. So this is very encouraging. And now we wonder, GAT3. Because we know in brain, it's not just GAT1, you have GAT2, GAT3, and then BGT1. You have other GABA transporter. So wonder, if you lose GAT1, is there any compensatory mechanism to the makeup, the loss of GAT1? And I will first, because from previous literature, we know GAT3 is very important, especially in the thalamus, just like Yulia talked about. So we look at GAT3 across the brain regions. We found, yes, in thalamus, you have much more enriched expression of GAT3. We have a quantification for that, about 4 to 7-fold of other brain regions, but you see some expression at cortex, cerebellum, and the hippocampus. However, at age 2 months old, in both mouse model, we did not see clear upregulation. So that means GAT1, if GAT1 is lost, it's lost. No compensation at this age. We also wonder the subcellular localization of GAT1, because we have talked a lot, my whole career, talk about the ER. So is a mutant protein inside the ER? And so I first wonder. So we look at both mouse models. The green one is GAT1, top 3 is a nuclear marker. And this is enlarged version. So the box region is enlarged here. So the red circle means I mark the region around the nuclear. And we did a quantification. We see something quite interesting. We see around the nuclear region, we see a clear ring, but only in Charlie's mouse model, but not in Maxwell's mouse model. So what does it mean? It's likely because at this stage in mouse model, we cannot distinguish from wild type allele versus mutant allele. So I think probably there are some wild type allele mingled here. And this is the quantification. We look at the non-somatic region, somatic region, the whole field. You see across all these different quantification measurements, you have a reduction. So in non-somatic region, you see very similar reduction in both mouse model. But in the somatic region, you see more reduction in Maxwell's mouse model. So I think this again suggests some protein is around the nuclear envelope, more in Charlie's mouse. And then also we look at the functionality of the GABA transporter. So we take a whole forebrain, we remove the cerebellum, and then we isolate a synapsome. We look at how much function remaining, because you wonder, what's the function level. So first we look at wild type, we look at the control, so untreated, and then also we treat with C966, and 7-1 is a GAT1 inhibitor, specifically block GAT1. We also use SNAP 5114, specifically block GAT3. So we get the idea about if you block GAT1, you have a huge reduction. But if you block GAT3, you have a smaller reduction, but more significant. So this suggests that in a forebrain, major contributions are made from GAT1. And then, this is the total. We measure the total without any blocker. And then here is a quantification. We only block GAT3. So basically, you remove the function from GAT3, and showing the function of GAT1. So you see almost a comparable reduction in both mouse models. And then this is the normalization. So at this point, we are comfortable. We want to see in a forebrain, in both mouse model, you have a similar reduction of GAT1 function. And then we did TGAB binding, because we know TGAB is a specific GAT1 inhibitor. And we have a beautiful work done from Roche. Our colleague Maria Clementia is here. We are so happy. And this work is done by Roche team. So here you can see very abundant expression, very stunning of GAT1, TGAB binding. The most abundant area is cortex and then deep brain nuclei, with showing a strong signal, the yellow here. And then we look at wild type and heterozygous in Charlie's mouse. This is the wild type, this is sagittal section, and this is heterozygous. Do you see across all these different brain regions? You see global reduction of TGAB binding. So it's consistent with our biochemistry study. You see global reduction of GAT1. And this is the quantification. Cross brain regions, you see very similar reduction. And similar in Maxwell's mouse model. This is the wild type. This is the little mate, it's male animals. So you see the reduction is almost identical to A2V. And now let's move on to neurobehavior phenotypes. Thanks to Yulia, just talk about here. We also have some data because we have done so much in multiple cohorts. We just selectively present few. So first, we identified both mouse models have an absence CA phenotype. Occasionally, maybe during handling, we observed a generalized tonic-clonic, and this is very rare. I think the major phenotype is absence. And this Maxwell's. So compare with Charlie's and Maxwell's mouse model. I want to say we noticed maybe the duration of this speculative discharge is a little bit longer in Chinese mouse model. And this is the EEG I got from 4 Chinese patients carry different mutations in SLC1A. So what is 4 different patients, different age from 2 to 8. So that this is the EEG we done from Chinese colleagues. They have a very typical, about around 2 to 4 hertz speculative discharges, which correlates what we have seen in mouse model, like 5 to 7 hertz theta activity. We have done pretty deep phenotyping in both mouse models, cost us literally $1 million. So we started from day 7 from retinal reflex and body weight, and now we seizure observation, and now we do better neurobehavior test and microdialysis. So we longitudinally monitor animal from 1 month to 10 months. So this shows the mouse weight, similar to Unia's data. We have done a lot. We show trend definitely in both mouse models show reduced weight when they are young, but then seems they catch up later on. For the retinal reflex, because this is my fifth presentation in this, in this symposium, I showed the video a few times, so today I did not show this video. But the short answer is both a mutant animal seems have a prolonged writing time if you put them upside down. For phenotyping, I just present a few on the time point. We found in both mouse models with home cage scan, we show robust phenotype in reduced travel distance, and from 1 month to 10 months. This for both mouse models, we see similar thing. Analysis is like home cuddle because we use home cage scan, so basically everything is automatic. The home cuddle actually talked with the software developer. This means basically hung. They have hung vertical and hung cuddle. So we saw robust phenotype. So the mouse have reduced activity in both mouse lines. We are also very happy to plot difference between these two mouse models, very subtle difference. So we know in 2-8 way, that one is associated with autistic phenotype. That's I have talked with physicians in China and Israel, and also like others. So this mutation seems some children have a very severe autism. But for Maxwell, at least Amber, I never heard that. So yeah, so we found like a repetitive behavior in this. Oh, actually, it's like next one. This one is showing sleep. So we saw increased sleep score in Maxwell's mouse model, but not in Chinese mouse. Now what we say in Chinese mouse have increased grooming, but not in Maxwell's mouse. So this is why we say actually we actually can tease out the difference between this very subtle difference with our behavior assay. Now we conduct microdialysis. 2 full-time employees, very laborers. We did that from 1 month to 10 months. We want to see how much GABA the actual level. We know what is mutant can cause defective GABA transporter, but what's the final consequence in the brain with time. So we did a microdialysis. Each mouse, we sampled 18. We had taken 18 samples. Every 15 minutes, we take a sample. We try to measure the GABA level. So this is the data. I just show you at the 7 months old. You see this Maxwell's heterozygous or male animal, and this is Charlie's. You see this is cumulative. We plot like a cumulative GABA level. Lower, this is like a different time sample this. In the lookout mouse model, in Bregina's paper, they used potassium chloride like 30 millimole to stimulate during the microdialysis. If you stimulate, you will see increased GABA. So we did a similar experiment. Instead of 30 millimole, we use 50. And so if you compare the GABA at that time point, if you stimulate with the potassium chloride, you see in heterozygous, you see a spike compared with the wild type. So we see suggests like a delayed clearance. And then we did like a cumulative GABA. If you compare with wild type and heterozygous, this Maxwell's mouse model at 7 months, you see increased GABA level. It's very encouraging, we see, because we identified for phenylbutyrate. So we treat an animal for 7 days, and then we re-measure a microdialysis. We found for phenylbutyrate can normalize GABA level. So this is, we feel very comfortable about that, and I'm very happy. That means we can use Ravictin, probably can modify the disease process because of this data. So the take-home message from my talk today is first, we demonstrate in both mouse models, GAT1 is reduced globally from what is brain regions we have sampled. We found out GAT3 is not increased in either mouse model, at least at 2 months old. We found GAT1, the GABA uptake was reduced in both mouse models in the forebrain synapsome. Later on, Corey and Melissa had done in the brain slice, we're not showing today. But I think we are confident the overall GABA uptake in the brain is reduced. And I will see robust phenotype of reduced travel distance and harm in both mouse models. And also we are able to see subtle difference in later stage of the mouse. And we found increased GABA level upon potassium chloride stimulation in mouse model, but we have not seen a clear response in Charlie's mouse. We found the increased cumulative extracellular GABA, which can be restored by 4-phenylbutyrate. So this is very assuring. With that, I'm very thankful to first Amber, the support, also all the people in my lab, and now when the behavior core, we have done so many tests, failed so many because there's nothing at the beginning until we have tried different things. And now we also very thankful to combine the brain and T-cell gene therapy or industry partner like especially UCB and then Roche help us generate the data with gigabin banding. Yeah. Thank you.